Chapter 13, Section 5, Regulation of Metabolic Pathways. I mean, look at this picture behind my head. When you look at this, it kind of gives you an indication of all these pathways that are going to be found in the cell that are used for regulating, that are used for catabolic and anabolic pathways, for signals, for all of it. And it looks like a mess. It's like trying to follow up an individual noodle in a bowl of noodles. Sorry, haven't eaten lunch yet. Thinking food, yum, noodles. But trying to trace things through this mishmash and seeing that sometimes some of these little tracings, well, guess what? They meet up. They become one together. Then they branch off again on their own as these signal pathways lead into and out of each other. And there's crosstalk. One regulates the other. One inhibits the other. One activates the other. So when we look at this and we're trying to discern it, and you're going to be doing this in lab, we need to keep in mind that you start with something, you end with something, and in between there you have this intermediate. Okay? You have the velocity, how fast, what's the rate of A to S? What's the rate of S to P? Okay, it's still similar to what we talked about last semester when we were talking enzyme reactions and enzyme pathways. Remember, just here, these enzymes happen to be kinases or something like that. When we see that S is neither going up, neither going down, this velocity, that velocity are equal. So as quickly as A is going to S, S is going to P. We think of this as homeostasis in the cell. When it's going so fast from A to S because V1 is so much greater than V2, A is being deplenished really fast, S is building up, P minimal. Not homeostasis, not good for the cell. So to maintain this, to keep this steady state, A to S, S to P, keep this nice, keep this going, what they call the flux. Well, you can change the number of enzymes, the enzyme controlling V1, the enzyme controlling V2. Or you can put some sort of a regulated catalytic activity on the enzyme. Speed up the enzyme, slow down the enzyme, depending upon what's required. And remember, timescales here for these reactions is going to be very variable. Measured in milliseconds, measured in days, depending upon what it is you want to do, what it is you want to discuss. I mean, when you look at enzyme activity, and this is, what we're going to see a lot of, when you see that's controlling the cell, some form of extracellular signal hits a receptor. The receptor kicks off a series of reactions. Those reactions could be, the dominoes, like I talked about in the lab, of kinases, boom, 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 knocking one over the other, resulting in transcription, translation, new enzyme. New enzyme causes different things to happen in the cell, or other things that are in the cell that have been held back now has an enzyme that is its substrate it can go and act on. All sorts of different things can happen. Those extracellular signals, hormonal, neuronal, say like acetylcholine, from neuron 1 to neuron 2 across the synapse. Could be some growth factors, could be some cytokines. Depends on what the signal is, depends on what the target cell is going to be, depends on the target cell, which signal cascade, which signal process you want to turn on or turn off. Those transcription factors I just mentioned, the transcription, got to get some transcription factors. These are what control the yay-nay if certain genes will be transcribed. Just because you have the genes in your genome doesn't mean you're going to have access to them at all points in times. No, there's going to be control elements, proteins that will bind to certain places on the double helix that inhibits DNA polymerase from coming in and making a messenger RNA copy of that gene. Or transcription factors are going to bind around the gene upstream from it, and they're going to recruit in polymerase and saying, here, make this one now. Turns out these transcription factors are going to be activated by signaling pathways, i.e., protein cascades, kinases cascades, that were activated because of some extracellular signal bound to a receptor. When we look at mRNA, because remember that's what transcription ultimately is going to yield, is mRNA, an RNA copy of a DNA gene that's part of your genome. The stability varies depending upon secondary structures. Think back to last semester when we were talking nucleic acids. DNA has the double helix that can help stabilize it more, but the RNA is a single strand. To get some stability in that single strand that nucleic acid strand, it will bend back on itself and form those stems and loops. Well, more stem loops and certain structures within those stem loops, greater the stability. Proteins themselves are not permanent. Just because it's one and done, it's made, it's always going to be there. No. Turns out proteins have a shelf life. Okay. Depending upon the cell, depending upon what the protein is, that shelf could be longer, it could be shorter. I mean, you look at this example here from table 13.8 from the textbook. Proteins in the liver have an average half-life of 0.9 days. That means all the proteins made today, right this moment, half of them are only going to last 0.9 of a day. Another day, 0.9 of that remaining 0.9 will be gone. Another day, 0.9 of that remaining 0.9 will be gone of the ones made today. Kidney, heart, brain, muscle, longer. Why so short in the liver? We got to remember what the liver does. The liver takes anything that's been absorbed directly from your gastrointestinal tract and filters it. Because remember, you absorb stuff from the food in your small intestine, large intestine, immediately goes to the bloodstream, and then travels directly to the liver. Do not pass go, do not go anywhere else, go to the liver. The liver takes the brunt of any of the bad stuff that you absorbed from your diet. So, hepatic cells are constantly getting damaged. It's their job. Their job is to take the abuse, which is also why you have a quick turnover of the proteins. You also have a quick turnover of hepatic cells. Brain, your brain's constantly working. Your muscles throughout your body, especially your skeletal muscles, not always working. I mean, certain muscles, they're working a lot, but not always. As I'm sitting here, there's certain muscles in my arms, certain muscles in my abdomen, certain muscles in my legs that just haven't flexed in a while because I haven't shifted weight. My weight haven't moved. But my brains keep firing. Okay? So, they're constantly working, constantly doing stuff. Gotta move. Same with the heart. Heart's always beating until it stops, and then, you don't really need to worry about replacing any proteins. But constantly beating, boom, 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 stresses the cells. Stress cells take damage. Stress cells, need to turn over the proteins. So there you go. Any damage to the proteins, they get turned over, get recycled, broken down to the amino acids. The amino acids go back to the cellular pool for the next polypeptide to be. A lot of times your cells will make enzymes because it has the time, it has the ability right this moment, but it doesn't actually need them. Or it needed them now, but it's not going to need them for a while. So instead of, wasting them, turning them over and then, once it's needed again, waiting for the minutes, the hours to make a new one, transcription, translation, post-translational modification, blah, 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 all of that, well, takes that enzyme and copies of that enzyme, and then they just put it away. Think about it. It's middle of winter now. It's winter. It's snowing. You got summer clothes. Nice light shirts, shorts, things like that. Are you going to throw those away? No. You're going to put them in a box. You're going to put them in a drawer, and you're just going to forget about them for the next few months until it's nice and warm again. Same thing here. Cells take those enzymes, going to need them again, and going to need them quickly again, but just don't know when. Put them in a vesicle. Put them in an organelle. Just let them set. We have what is referred to as the proteome. Proteome is the collection of any and all proteins in a cell type. The proteome changes as certain proteins are made, certain proteins are degraded, and what outside signals, stresses, are upon that cell. Well, if you have the proteome, you also have what is referred to as the transcriptome. What mRNAs are present? Okay. There's the baseline mRNAs, part of that homeostasis. Okay, here's just what the cell needs to be a cell, to be a skin cell, to be a liver cell, to be a neuron. And then there's the mRNAs that are made because, hey, some outside signal. Cell damage, cell stress, oxidative stress, change in pH, things like that you're reading up on, hopefully, in lab. So, what messenger RNAs are present in the cell will change. Same with what they call the metabolome. What are the metabolic reactions that are occurring in the cell right this given instant as the cell is just being a cell? No stress, no nothing. Here's what you should see at the bare minimum for a liver cell, for a kidney cell, for a skin cell. Something happens, an injury. Something happens, your liver cells just got exposed because you ate some weird chemically laden food, blah, blah, blah. The metabolism is going to be altered. Well, what's the new altered state? What's being done there that wasn't done for the homeostasis, the baseline? That's what we get to study. When you look at, we've done this study like for bacteria like E. coli. And, here is the metabolic functions of E. coli that's growing on glucose. Here's where all the metabolism is focusing on, the majority of it is focusing on things that are needed for glutamate metabolism. But then, hey, look, you start to see stuff like fructose 1,6-bisphosphate, hexosphosphatases, things like that. Oh, hey, wait a minute, that's glycolysis. Oh, hey, look, the building up of our triphosphate starts to kick in a bit. Ooh, the making of certain amino acids, the making, the catabolic, anabolic reactions for certain amino acids, all because this E. coli is now growing in a glucose-rich environment. That's metabolomics. Now, if you want to regulate an individual enzyme, we're not looking to push or pull the overall metabolism, the overall, cascade pathway, but you want to just regulate just one. Well, first off, you can go by concentration of substrate. Remember, enzyme is go, go, go. You want to slow down the production of a product by that enzyme? Give it less substrate to work on. Okay, that's one way to do it. Have somewhat of an allosteric effector. Okay, remember allosteric effector, different than our competitive inhibitors. Remember, competitive inhibitors worked at the active site. The allosteric bound somewhere else on the protein, on the enzyme, and change the overall shape of the enzyme. Change the shape, lose the function. That's if you're an allosteric inhibitor, or you could be an allosteric activator if the native shape is like this, but the allosteric activator binds, and now it changes to the true shape of the enzyme that has the active site there. Boom, now it can go. Allosteric activator gets popped off, goes back to that native have no function shape. That's a great way to do it. There could be covalent modifications, the addition of a methyl group, the addition of, small polypeptide. And then there's binding of regulatory proteins. Could be, a vitamin, could be some form of a cofactor, needs to be there. Just there to help with the taking of the substrate and making the product. So these are ways to regulate. And when we look at the Michaelis-Menten Kinetics, because remember, these enzymes, we talked about this last semester when we look at, there are ways to measure the enzyme, and when you look at, the Vmax and all of that, but if you come closer and closer to the beginning and you look at what's happening here at the beginning at the smaller concentrations below Km, you'll see that the activity drops off the lower the substrate concentration because the enzyme hasn't reached peak yet. So we're below Km, because remember Km is when half of the Vmax. And when concentration is so much lower than Km, it's almost a near, our kinetic rate now becomes dependent. More S, higher rate. More S, higher rate. Unless we're approaching Km, or we're beyond Km and now we're at Vmax. Now you can blow out S all you want, and there's only so much the enzyme can do. So if we're at a constant enzyme, constant E, but our concentration of S is so low, well, now you can play around. You can vary things, and our rate will also be highly variable. Those allosteric effectors, like I told you, they can slightly alter the shape of the enzyme, make it so things happen or make it so things don't happen. So that hyperbolic kinetic, where it was just up and over, now becomes more of a sigmoid, like you've seen in physics. So we can make things happen. We can turn things off by having, increase, decrease of activity based on the presence, absence, or how much of the allosteric effector is there. And a lot of times, what we're going to see is that this covalent modification that I talked about a couple slides ago, where I said add a group, subtract a group. Well, it's going to be phosphate, an inorganic phosphate from ATP. Remember those kinases we talked about in the lab that you're now doing research on? P38, CJUN, things like that. Well, in its native form, it's just there. Add in a phosphate group, and now it's active. Remove the phosphate group, using a phosphatase, goes back to being inactive. We see that enzymes can be regulated. Transcription, whether or not yes or no, whether or not the messenger RNA that will be made. No RNA made, no protein. Yes, the RNA is made, got to be a protein. Once the protein, the polypeptide's made, then whether or not there's going to be an allosteric or some form of an addition, like the phosphate group from the last slide. So this gives us levels of control, us being living cells. And then the metabolic regulation, okay? Just because you have the enzymes there, you have the pathways there, doesn't mean you need to use them, doesn't mean they need to be active. Sometimes you need them just to be there and just wait. Other times, ooh, need them now, need them low level. Okay, need them now, but we need them high level. This is where regulation and control come into play. So if you have a pathway, start at A, here's the initial substrate, D is our final product. Yeah, we can. The enzyme 1, 2, 3, yes, we can control at the transcription level while they're 1, 2, and 3 is being made. But which of these steps do we want to control? Using some allosteric mechanism, some other mechanism. Well, it turns out we want to look for the ones where equilibrium is not always optimally done. V going from A to B is our 10. V going from B to A, 0.01 is its rate. Wow. That's quite a big difference. Whereas you look at these others, 200, 190, 500, 490, these are quite close. This, not so much. So if we could find a way to control this, because it's just, this reaction just wants to go A to B so easily. Whereas these reactions, B to C, is just as likely to happen as C to back to B. Okay? This would be the best point of control. And we see this when we look at the mass action ratio. Q equals concentration of the products divided by the concentration of the substrates. Yes, this kind of looks quite similar to our K prime equilibrium we talked about in section 1 of this chapter. But you remember the K prime equilibrium had C to the C power, D concentration of D to the D power divided by concentration A to the A, blah, blah, blah. Okay, we're not taking all that into account here. We're just looking at concentration, boom, boom, boom. When Q and EQ, K prime are 1 to 2 orders of each other, the reaction is going to be an equilibrium. So on those previous where, 500, 490, 200, 190, yeah, really close there. Whereas the one that was 10 and the other one, one way and 0.001 the other. So you start going and you're looking at, what it takes, what's inherent within certain enzymes, certain reactions they will control. Hexokinase, okay, adds a phosphate group to glucose, first step of glycolysis. KEQ, 10 to the third. Q, 2 times 10 to the negative 2. Wow. In the liver, 8 times 10 to the negative 2 in the heart. Is this anywhere near equilibrium in vivo? No. You take other ones like, let's go with glucose 6-phosphatase. KEQ, 8.5 times 10 to the 2. Q in the liver, 1.2 times 10 to the second. Huh. Pretty darn close. 10 to the second, 10 to the second. Are they at equilibrium? Yes. Now, does that mean that this reaction can't, that this one's controlling can't happen? No. It just means this isn't going to be a great one to go through and try to regulate. And ATP, okay? ATP concentration. When it comes to enzyme activity. As ATP levels drop, well, enzymes slow down. Enzyme activity becomes less. ATP seems to be one of our mechanisms not only for activating but for controlling these kinases, these enzymes, activity, their ability to work. Different kinases, different roles. Adenylate kinase catalyzes production of AMP in the reaction of 2-ADP to AMP and ATP. Strips off one of the phosphates here, a diphosphate, to make a TP and a monophosphate. Why would you want to have that? Well, again, you need triphosphate to power certain things. And if the oxidative phosphorylation in the mitochondria is not keeping up, it happens to be one of those cells that doesn't have creatinine. Well, here's what you would do. AMP activated protein kinases. These protein kinases look for this level here. When ATP is so much higher than AMP, high number here, high ratio, these are not activated. When this number gets lower, I mean, it's never going to get too low, but when it gets below a certain threshold, that means the AMP levels are going up, ATP levels are going down. These guys get activated, and different metabolic activities will kick in so that the ATP levels can be taken back up.